Guys, today we are going to cover our very first chapter of AS biology. So, our very first chapter is about cells. So, this is okay. Let me also give the subject code. It's 9700 syllabus. So, in the first chapter, in the very first chapter, we actually see about cells. So, what are cells? Cells are basically the very smallest functional and structural unit of any organism. So it is the smallest functional, this word is very important, functional plus structural. So functional means it refers different types of functions such as protein synthesis, respiration, or any other kind of function, and structural means it has a specific concrete structure. So smallest functional and structural unit of any organism. Okay, it is also known as a unit of life, and cells can actually undergo division. So we have seen cell division also in our O levels. We've seen mitosis and meiosis. So those are the cell divisions. So yeah, and they can be passed on from one generation to the next. So we have learned about inheritance. Anyways, so that's a basic definition of any of cells. Okay, the next thing we need to deal with in this chapter is about microscopy. We know that we have to know about two microscopes. One is the light microscope and the other one is an electron microscope. So we need to know that light microscope uses light as a source of radiation or as an illuminating source, and electron microscope, as the name suggests, makes a use of electrons as the radiation source. Okay, radiation source. So these are the two differences. These are the two types of microscopes which we are supposed to learn about in our AS level. So at first we need to looking at light microscope. There are two measurement units we need to know about. One is the eyepiece graticule, and the next thing we have to know about in light microscopy is the scale micrometer. Okay, so we are going to learn about these two things. So what is the eyepiece graticule and what is the stage micrometer? Sorry, that's supposed to be staged. Okay, stage micrometer.